We're going to talk a little bit about the eye. First, we're going to talk about how the eye focuses on light. Then we're going to focus in a little bit on how the light is processed and changed into action potentials in our brain, so our brain can figure out what's going on through our eyes. So this is a rough drawing of an eye here, and with that, each one of these points is a point of where light is being sent off of an object. And when that light comes in contact with our eye, the first point it's going to hit is the cornea. And the cornea is the initial point of refraction. So it's going to be the first point to bend light. But then it's going to hit the second part of the eye, and that's the lens. And the lens is what helps fine-tune the focus of the light, which then helps it focus it in on the retina, which is this back part, is the retina. On the retina, it's loaded with these things called rods and cones. Rods help us see a little bit more black and white and in darker settings, and cones allow us to see color. There are three types of cones. The one helps us see green, one helps us see blue, the other one helps us see red. And through the combination of those three colors, the millions of cones that we have will help us then piece together what we see through and what colors we see. And so if we have this closer point, since it's a closer point, the light is going to be bent more than when you're further. With this further point, the light's not going to need to be bent as much. And so the way to make up for that and help us allow to focus on different points and different depths, our lenses has a really cool mechanism to it. So the lens, so when you're trying to see near, you have these muscles called the ciliary muscles. And when these muscles contract, they end up loosening these ligaments, your suspensory ligaments. And when those suspensory ligaments are loosened, the lens will become rounder, which then allow a greater refraction. It curves the light better to then focus in on the retina. But when you need to see further, the ciliary muscles will loosen or relax, which then tighten these suspensory ligaments, causing the lens to flatten and not bending the light as much, allowing you to see a little bit clearer. And so within the eye, there's a few different key points to it. And in the back, we will have a couple different sections here. And so there's this place called the optic disc, and that's where there's no rods or cones. And that's where your nerves are going to run through to create the optic nerve leading to your brain. And you have this place called the fovea, which has a very high concentration of cones. And that's where most of your light's going to be focused in when you're trying to see. And so the mechanism, which we show right here, is very intricate. Okay, so the way we process light here, this is a photoreceptor, this first cell on the left, and it has this little protein, a G-coupled protein receptor, and within it has this molecule called 11-cis-retinol. When the photons come in, it breaks that bond and reforms it as a trans bond. And once it has that trans bond change, it causes the G-coupled protein receptor to release its alpha subunit. And when it releases that, it activates phosphodiesterase, this little red box here. Once that's activated, it actually deactivates CAMP, which beforehand was activated. And if CAMP's deactivated, it closes this ligand-gated sodium channel, which doesn't allow an action potential to occur. So the little red lines is when light actually is entering into the photoreceptor, and it doesn't allow this action potential to occur. If that action potential doesn't occur, calcium channel doesn't open, and neurotransmitters are not released, which is a good thing. Because if we have neurotransmitters released, it then activates this mGlu-R6 G protein coupled receptor, which then releases alpha subunit, which opens. It will open this TRPM1, and this opens when there isn't glutamate. So, but when there is, this reaction does not occur. The mGlu-R6 is deactivated. And so when the neurotransmitter doesn't come, because of the light that hit the photoreceptor cell, this calcium channel is allowed to open, allowing influx of calcium, causing that depolarization and causing the release of glutamate within this middle cell. And when that occurs, it does release glutamate to these neurons, and when glutamate is released, it interacts with AMPA, which then opens up AMPA, causing an action potential to occur, causing a signal to go to the brain. And there's a special type of cell. There's two types of cell. That's a on-center photoreceptor, and an off-center photoreceptor. There would be an AMPA channel here as well, and it would do the opposite effect. So when it does that, it sends, when you do receive photoreceptors on the off-center, it sends the action, it doesn't send the action potential, it doesn't release neurotransmitters, which then doesn't allow this action potential to occur, which doesn't send a signal back to the brain. Finally, I want to talk a little bit about this on-center, off-center sort of deal. So on the on-center, like we said, there's the mGlu-R6 channel, which when glutamate is released, then doesn't allow, or then glutamate is released, which then allows that, doesn't allow that action potential to occur, versus the AMPA channel, which does. So right here's that AMPA channel, here's that AMPA channel on off-center, but on on-center, the AMPA channel is still here, but the GLU-R6 is here, which turns off the action potential for when glutamate happens or is released. So these are good examples of what happens with your visual fields. There are lots of these little visual fields, and they all, when you focus in on the fovea, each visual field is directly connected to one photoreceptor, which is then directly connected to one neuron, but off the fovea, it's more concentrated, and it looks like, this is very simplified, there's only 3 photoreceptors, but there would be the center photoreceptors and then these peripheral photoreceptors, which then connect into the center photoreceptors through lateral cells. So a very interesting thing here is that within these visual fields, we have this on-center and off-surround. When light is hitting all of it, the on-center doesn't release glutamate, which then allows this action potential to occur, which sends a signal. And that happens, let's simplify and say 60% of the time. While these off-surrounds also are active, they allow the release of neurotransmitter here, which then inactivates here. It sends a signal into it, shuts things down, and doesn't release the glutamate. And so now we have this whole section is absorbing or sending a 60% signal to the brain. In comparison, if you cover up a section of your visual field, that one side or one peripheral section of this visual field is covered, the covering actually will then not have this happen, which won't shut down a portion of your cell here, of your photoreceptor, while this side still is. So now you have about an 80% effect, represented by these lines up here. But then when you cover up the center, it shuts things down. You're not getting light, which then doesn't change anything and doesn't release glutamate, which then doesn't allow this to occur. And so you're only getting about 20% from this other side, allowing things to occur. And then if we look at the off center here, the exact opposite happens. And so when you have it fully absorbing light, it's 40% occurs from the outside sections, but then when it's completely covered, only the center would be active, allowing the 60% to occur. But when it's partly covered here, the center isn't active, but only one side is, which allows it to occur 20% of the time. And when the center is covered, because an off center means it would be sending that signal, but the glutamate would hit it and allow this action potential to occur and go because of the AMPA channel here. And that is it.